Hello everyone and welcome to my channel. Thank you for joining me today. My name is Naza and we are going to dive into a topic that is pretty fundamental to microbiology. Our topic today is bacterial taxonomy and scientific nomenclature. So let's start with a question that you have probably wondered about before. Why and how do scientists group microorganisms? Have you ever thought about that? Well, it is not just for fun, it is to create a universal language that helps us to understand and also to communicate about this tiny organism. So let's get into the specifics of this system. To get a clear picture of how this all works, we are going to cover three main things today. The first one, we will look at the history of microorganism naming. Second, we will go over the basic rules of nomenclature. And finally, we will see why this system is so important for the scientific community. So now let's go back in the time to the 1700 to the year of 1700s. We have to talk about this guy, which is Sir Carl Linnaeus. He is often called the father of modern taxonomy. He was a Swedish botanist who developed a system for naming plants and also animals. So he is the one who gave us the binomial system, which uses two Latin names, a genus and also a species. So these scientific names might sound complicated, but they actually come from four sources. So the first one is descriptive, second is the scientist names, third is geographic places, and the last one is organization. So for example, for description of organism, for example like Staphylococcus aureus, which means grape-like cluster of spears, golden in color, and also they can also be named after scientists, for example like Escherichia coli, which named after Theodore Escherichia, and also sometimes the name comes from a geographical place like Blastomyces brasiliensis from Brazil, or even from an organization such as Legionella, which was first discovered in American region. So this is the taxonomy of bacterial nomenclature. So it is the science of classifying organisms to construct internationally shared classification system. It is also known as Linnaean system. So we have Escherichia coli, for example here. So for E. coli, the kingdom is bacteria, the phylum is Proteobacteria, the class is Gamma Proteobacteria, order is Enterobacteriales, family is Enterobacteriaceae, genus is Escherichia, and species is Escherichia coli. So now, for the fun part, the rules. There are a few key guidelines we follow to make sure everyone is on the same page. So we have 9 rules here. So rule number 1 is to use binary names. So every organism has a generic name and a species epithet, like Escherichia coli. Rule number 2, capitalization. So the genus name is always capitalized, but the species name is never capitalized. For example, as you can see here, Bacillus anthracis, the word B should be capitalized, but the word A for anthracis is not capitalized. Rule number 3, italicize. When you are typing, both the genus and species name should be in italics, and if you are handwriting them, you should underline them instead. Rule number 4, abbreviations. Once you have used the full name once, you can abbreviate the generic name to just the first letter as long as it is not confusing. So for example, like Escherichia coli, it can be written as E. coli. Rule number 5, common names. So common names like Streptococcus are not capitalized or italicized, similar for Brucella. And then rule number 6, subspecies and also serovars. These have their own rules. For example, Salmonella enterica serova typhimurium. So as you can see here, how we write Salmonella enterica serova and typhimurium. Rule number 8, plural forms. Just like in English, some words have a special plural form. So for example, the plural of streptococcus is streptococci. Rule number 9, which is the last one, the word zero versus O. This might seem small, but this is very important. So the letter O and the number 0 look alike, but they are not same. So for example here, E. coli antigen O157. So it's not E. coli 0157. So with all these rules, you might be asking yourself, why do scientists even bother with a proper naming system? Well, this is a great question, and there are 4 key reasons to answer this question. So number 1, it helps us to organize the huge amount of knowledge about organism. Number 2, it helps us to make predictions and frame hypotheses for further research. Well, number 3, it allows for universal communication between scientists. And finally, it is crucial for the identification of organisms. So the proper nomenclature provides a standardized system for clear and accurate identification, which is vital for effective communication and research in the scientific and medical communities. So in summary, taxonomy and nomenclature give us the essential tools to classify, to organize, and also to discuss the world of microorganisms. And also it is a system that has been developed over centuries to ensure clarity and consistency in science. Thank you very much for your attention. Hope to see you again in the next session.